Welcome to Click for IB Biology. We're looking at classification and cladistics. There's a PDF of this presentation that's linked in down below in the description. You can download that, add your own notes, and then when complete, why not take the 10-question quiz and check out your answers to secure your understanding and learning. Classification, of course, is the father of classification is Carl Linnaeus. Back in the 18th century, Carl Linnaeus was classifying the overwhelming diversity of living things into categories. His categories had a hierarchical structure of kingdom, class, order, genus, and species. This classification, of course, is the roots of the word taxonomy lie in taxis, which is to arrange, and nomina gives us the method. So the method of arrangement is seen in this hierarchical structure of the kingdom, class, order, genus, and species. And of course, you're familiar with the idea that each species has a binomial name, another aspect of Linnaean construction, in which we specify the genus with the uppercase letter and the species epithet with the lowercase letter. So this is ours, of course, Homo sapiens, and this is all based on the morphological features that Linnaeus was able to observe on the exterior of the organisms. And so this was the predominant mode of classification. There was no implication in Linnaean classification of evolutionary relationship, and we can see that when we look at Linnaean groups of reptiles, which are based on having scales, eggs, and being cold-blooded, ectothermy. That makes you a reptile. And if you're a bird, then of course we have feathers and endothermy. And so we have these two distinct groups. But there was never any implication in Linnaean classification about the evolutionary relationship between these two groups. So then when we fast forward to the early 20th century, Will Henning introduced the concept of cladistics diagram as illustrated here. And this very much is based on the idea of showing the evolutionary relationship between groups. And you can see immediately that in this particular example of a cladistics diagram that the crocodilians and the birds are very closely placed in this diagram, and this proximity in the diagram would suggest that they have a common ancestor and that they constitute an evolutionary group, as distinct from the turtles with snakes and reptiles, which are up here. And so this is reconstructing the diagram, the classical diagram of Linnaeus to show the evolutionary relationships. And of course, these diagrams are called cladistics diagrams. So let's look at the features of cladistics diagrams and how they might show evolutionary relationships. At the root of a cladistics diagram, as indicated here, is the last common ancestor of all the groups which are shown here, which are just theoretically shown as A, B, C, and D. And the groups at the terminal points of the cladistics diagram are called taxa. And they may be current species, or they may include extinct species. Now, the concept of a clade has two parts to it. One is the idea of it must include the common ancestor, and the second part is that it must contain all of the descendants. So we'll call that part two. Can I emphasize it says all of its descendants? This is an important feature. The splitting points in the evolution of groups, because that's what we're trying to indicate, are known as nodes. So these splitting points perhaps could be related at some point to speciation events. The branches are connected. Notice that in this particular diagram that the branch lengths here and here, and here and here, they're all the same length, and that's because they do not represent evolutionary time. So in the pure cladistics diagram, this does not include evolutionary time. Let's take a look at some theoretical aspects of cladistics diagrams, and here what I have in mind is that those of you watching this video are probably facing examinations, possible multiple choice questions, in which they ask you to identify a legitimate clade. So remember that the clade must include a single ancestor and all of its descendants. So let's take a look at this. So if we circle this group here, are C and D a clade? Well, it includes the descendants C and D, and we have this node point here indicating this is the single ancestor. So if we cut the cladistics diagram at this point, then this is indeed a genuine clade. So that one's a good one. If we come over here to this diagram here and we select this as our clade, let's examine the criteria. Does it have a single ancestor? Yes, that would be here. Are all the descendants included? Yes, there is only one, and so again it meets the criteria for a clade as represented by the circle surrounding D. And notice that once more we only made a single cut to our diagram in order to achieve that. Now if we look below, and let's just begin by circling A, B, C, D in this manner here. And so within my blue line, is this a clade? Does it have all the descendants? Well, yes, it does. That's A, B, C, and D. But notice that it requires two cuts, leaving this root here and this root here. So that is not a single ancestor. That would be two ancestors. This is not a legitimate clade inside the blue line. Across here on the right hand side, if we were to cut the clade here and here, and then let's just circle A, B, C, D, A, B, C rather, like so. Is this a clade? So notice that whilst there is a single ancestor, so we're okay on the first criteria, it has not included all the descendants, and so it fails the test of a clade. In very general terms, it is the case that if you can create your clade with a single cut to the tree, then this is a legitimate clade. If you have a double cut or more, it is not a legitimate clade. Today, clades are enhanced by the addition of molecular data rather than simply basing them on pure morphological structures. Clades technically still accurately depict the relationship based on shared derived features, but there is a transition has been made in cladistics to what is called a phylogenetic tree, and the principal feature of a phylogenetic tree is an attempt to show the evolutionary relationships, including both genetic and morphological data, and that then, as a consequence of that, notice that these branch lengths are different, and what they're trying to do is to indicate evolutionary time and the events which occurred in evolutionary time. And so you can see that this splitting event here occurred theoretically before this one here, and in order to actually achieve these phylogenetic diagrams, it's necessary to obtain molecular data with a time component, and this time component is what emerges from the comparisons which are possible in phylogenetics. To do such comparisons, one has to obtain gene sequences from the various species that we're looking at, or the various groups that we're looking at. And if you are not obtaining these by experimental data, then one could resort to looking at genetic databases such as GeneBank, and by obtaining those, we get our sequence acquisition. So we have the sequences of genes that we want to compare between our groups of organisms that we're looking to study. Having done that, what we then do is we carry out multiple sequence alignment, which is today is performed by software. There are hand-based ways of doing this and applying the technique of parsimony, but these are very, very time-consuming. But today, with applications such as Clustral Omega, this can be done very, very quickly online. So students could go to the National Library of Medicine, the gene bank, and obtain gene sequences, and then deploying Clustral Omega, download the software and use that, you can begin to compare the gene sequences. And that part of this mechanism includes analysis of substitution models, sometimes performed by more complex analysis called the Jukes-Cantor models. And what we're doing with substitution models is comparing in the base sequences where those differences actually occur and the number of differences. Once this has been complete, further software will actually build your phylogenetic tree, which includes a time component. Software such as Mega allows you to do that. I've put the links to these on the left-hand side. Now, in advanced studies of evolutionary trees, it's then necessary to evaluate the reliability of your trees, in which case you would turn to statistical mathematical analysis, such as Bayesian posterior probabilities, but this is far beyond the syllabus that we're currently studying, but may feature in the work of students who go on to study genetics and evolutionary biology at this level of complexity. And so the outcome of these are phylogenetic trees which include a time component. And this is based on the concept of the molecular clock, which is underpinned itself by the idea of a constant rate of mutation. And we use those mutation rates to calculate differences, the differences, of course, emerging from here. And then we use that to calculate the time different branching points at our nodes. So this is an introduction to cladistics diagrams and cladistics analysis. The links appearing now here are to one of the great breakthroughs in the application of cladistics in the three-domain structure of life. There's the link to the channel. Please do subscribe. And on the right-hand side, there are the playlists. And if nothing else, give this video a like. Thank you for watching.